Thank you, Ronald. Welcome, everyone. So good morning or good afternoon, depending on your region of the world. Today I will talk to you about antisense approaches for the treatment of Duchenne muscular dystrophy and the recent advances also to address DMD brain comorbidities. So, as you know, RNA-based therapies have made tremendous progress over the past few years, and this is particularly true for the treatment of neuromuscular disorders and Duchenne muscular dystrophy in particular, because there are already 4 ASO drugs approved in the US and in Japan for the treatment of DMD, and there are many more currently under clinical evaluation. So today, I will briefly remind you the concept of exon-skipping therapy for Duchenne muscular dystrophy, and then go through the preclinical development of an alternative chemistry of antisense oligonucleotides, which are the Tricyclo, and in particular development of lipid-conjugated Tricyclo compounds. Then go through some of the remaining challenges, in particular the limited amount of DMD transcripts, which is a limitation in this strategy, and also the CNS targeting and the possibility of addressing brain comorbidities. So, as you know, Duchenne muscular dystrophy is caused by mutation in the dystrophin gene, also called the DMD gene. This is the largest human gene, and it's spread over 2.5 million base pair. And it encodes various isoforms of dystrophin that are represented here with the red arrows. The most commonly known is the full-length muscular isoform, which is also called DP427 for its molecular weight. And this protein plays an important structural role in the muscle fibers as being part of a dystrophin-associated complex. And the absence of dystrophin in patient induces degeneration of the musculature and unfortunately, premature death of patients. So dystrophin is expressed as various isoforms. As I mentioned, there are several full lengths, but also shorter isoforms, and some of them are expressed in the brain, and I will come back to this at the end of my talk. So the idea of the exon-skipping strategy for Duchenne muscular dystrophy actually emerged in the 90s, so it's been a while, and it was initially considered because most of the mutation that are found in DMD patients are actual deletions of one or several exons, and this is represented here with a deletion of exon 50. And the deletion of exons most of the time disrupt the reading frame, as it is the case here, where exon 49 and 51 are not in frame. And so you have the appearance of a premature stop codon that stops the translation, and the protein is truncated and non-stable. So the aim of the exon skipping is to actually restore the reading frame using antisense sequences that will mask some important splicing signals in order to skip an additional exon, restore the reading frame, and allow the translation to continue. And that will end up in the production of a protein that's slightly deleted internally, but still functional. And it's the type of protein that's found in Becker muscular dystrophy, which can be much milder than Duchenne. So the first oligonucleotides that were developed for Duchenne muscular dystrophy and the first clinical evaluation results were reported already in 2007. There were lots of different chemistries, or at least several chemistries evaluated for Duchenne. The first one were the 2'-O-methyl. And despite showing some encouraging results in initial trial, all the programs got terminated because they couldn't demonstrate therapeutic benefits, and that was also associated with some side effects. However, the PMO that were tested at similar time received FDA approval. The first one was eteplirsen, targeting the exon 51 in 2016. and some others after targeting other exons. There has been a lot of other developments, and I've noted here only one of them. For example, the StereoPure initial compounds that were also developed by Wave, and some of the initial programs with these StereoPures got terminated, just to illustrate the challenges that are associated with this disease. And also that the currently approved chemistry only restore a few % of dystrophin protein. So there are a lot of challenges still ahead. And in particular, we still need to improve the efficacy of the strategy and in particular improve the targeted delivery to ensure a higher bioavailability of these compounds, and target all the affected tissue, which is not little considering a muscular dystrophy. You need to target all the skeletal muscle, but also including the heart and the CNS in the case of Duchenne. So there are a lot of new developments to improve this technology, and you are, of course, heard about peptide conjugating oligo, antibody conjugated oligo, compounds that facilitate the endosomal escape, also some vectorized approach that we'll very shortly touch upon today. And so there are many reviews out there where you can see all the improvements. So today I will focus on one alternative chemistries that we've worked on, which is called the Tricyclo-DNA. So this is a chemistry that was created by Christian Lehmann at the University of Bern. And this is an interesting conformationally constrained oligonucleotides. It has specific properties, in particular an increased stability towards degradation and an increased RNA affinity, which makes this particular chemistry particularly interesting to be a therapeutic antisense oligonucleotide. So nowadays, these oligos are exclusively produced by Sky Therapeutics, which is also based at the University of Versailles. And several years ago already, we've demonstrated the superiority and the potency of this chemistry in the MDX mouse model. So this is the most commonly used mouse model for Duchenne muscular dystrophy. It carries a nonsense mutation in exon 23. So a stop codon appears prematurely in exon 23. Fortunately, this exon is in frame. So if we skip this exon, we can remove the stop codon and induce the synthesis of a almost fully functional protein. So at the time, we compared the tricyclo with the two chemistry that were used in clinical trials, the PMO and the 2'-O-methyl. And we were able to show that the tricyclo showed very high potency, at least 5 to 6 times higher potency. And this was associated with therapeutic benefits in the dystrophic mice, so restoration of the muscle function, respiratory function, cardiac function. So overall, they did show a high therapeutic potential, but what about the safety profile? So they initially showed a very encouraging profile. Like all the phosphosyrate oligonucleotides, they tend to accumulate quite a bit in the liver and kidneys. So we also checked for urinary biomarkers and in particular the kidney injury biomarkers. And you can see here that after 12 weeks of treatment in the dark gray bar, we could detect a moderate increase in renin and KIM1 biomarkers. So this is quite typical for PS, antisense oligonucleotides. However, we wonder at that time if it wouldn't be better to have an ASO that would be PS-free. So we investigated the possibility of lowering the PS content in the tricyclo oligonucleotides. This was particularly attractive, an attractive strategy for tricyclo because they are very stable, even in their phosphodiester form, so in their PO version. And the PS for tricyclo was mostly useful for biodistribution and serum protein binding. So we evaluated lots of different tricyclos, so always the same sequence targeting the exon 23. Having them from 0 0 PS link to 100% PS link. So this is a full phosphodiester, and this is a full phosphothyroid. And you can see here that after injection in mice, there's a clear contribution of the PS. So this is the tissue accumulation of the accumulation of the compound in tissues and the different muscle tissue, but also liver and kidney. That clearly correlates with the percentage of PS, and that correlates also with the efficacy. So the compounds inducing the most exon skipping are clearly the compound with the highest PS content. And that's not a surprise considering the serum binding. So this is the serum, the plasma protein binding of the oligonucleotides. So clearly the fully phosphothyrated oligo bind a lot of protein in the plasma, whereas the full phosphodiester do not bind much protein. So reducing the PS content is possible, but it clearly affects the biodistribution and the efficacy. So with this in mind, we had to, if we really wanted to develop a tcDNA PS3, we would need to compensate for this. So we really wanted a TC full PO that has the advantage of having minimal interaction with protein, but also no stereoisomer. So we compensated for the absence of PS using fatty acids. And here I'm going to show you some results using the palmitic acid. So we first compared the PK profile of conjugated and unconjugated compounds. So both full PO or full PS that would be conjugated to the palm or not. So we immediately saw that the palm compound clearly had a longer circulation. So on average, the area under the curve was increased by 10-fold approximately. So the palm tcDNA PS was 10-fold higher than the naked PS, and similar for the PO. And that correlated with a widespread distribution of the compound and a higher distribution to the muscle tissue, as you can see here. So here's the amount of tissue, the amount of compound that we detected in the different muscle tissue, skeletal muscle and heart. And interestingly, that also correlated in efficacy. We were particularly interested in looking at the previous generation in black, which is the unconjugated Tricyclo-PS, with the new generation of PO conjugated to the palm. And we could see that the palm PO tend to induce even higher exon skipping than the previous generation of the TCPS. So we then did a long-term treatment at various doses in MDX mice, so treating the mice with either 2, 4, or 10 micromole per kilo for about 3 months to look at dystrophin rescue and also functional outcome. So we were able to show a dose-dependent, you can see here with the palm PO, the palm PS, dose-dependent rescue of dystrophin in the muscle tissue. It was always higher with the palm PS, except in the heart, where the levels were actually very similar. And you can also see this on the immunostaining here, looking at the dystrophin expression. And of course, dystrophin expression was associated with functional improvements, such as the specific force, resistance to fatigue, respiratory function. But what we wanted to look at was the safety profile of these compounds. So we first did an in vitro screening to look at, for example, complement activation or coagulation pathway that are often impacted by PS content. And we could see that the palm PO did not activate the complement in mouse or human serum, but also did not affect the coagulation pathway. So there's no increase here in the prothrombin time or here in the activated partial thromboplastin time, which is impacted by the tcDNA PS. So overall, in vitro, it looked like the palm tcDNA PO compounds presented a safer profile than the PS. In vivo, when we after the injection, we had not noticed any any acute response, and there were also no changes in hematology or inflammatory response or complement activation in rodents. After the long-term treatment in serum biochemistry, there was no difference either, except, the only difference that we saw were actually positive effects from the treatment, because in dystrophic animals, you have elevated CK, you also have elevated transaminases that tend to go down with the benefit of the treatment. But what about the urinary analysis that we had seen elevated with PS before? So here, for the general urinary biomarkers like total protein or albumin, there was no elevation as opposed to the PALM PS. And again, in the specific acute kidney injury biomarkers, this time, even after 3 months of treatment, there was no elevation. So the PALM tcDNA PO definitely showed a safer tox profile than the PALM tcDNA PS. So we decided to continue with this new generation of lipid-conjugated that showed really a higher therapeutic index. And based on these results, so we developed a lead compound of PALM tcDNA PO to target the exon 51 of the DMD gene in partnership with Sky Therapeutics. And we ran some preclinical studies in non-human primates, and that revealed a very favorable tox profile, as we had seen also in rodents, but also a very efficient exon 51 skipping, as you can see here, where the lower band corresponds to the skipped product, because the oligo has a full match with the primate sequence. So after only 4 injections, we had a very efficient skipping. So based on these results, Sky Therapeutics has launched a phase 1/2A clinical trial in June last year. And obviously, I cannot comment on the trials, but all the phase 1 is completed. And what I can say is that in March this year, they received the authorization from the EMA to initiate phase 2A. So all the patients that were involved in the phase 1 will now progress to the phase 2A. So these are very exciting times for us. And although the phase 2 will take about 32 weeks, so we won't have results in a little while. This is quite exciting. So moving to the next part of my talk and about the remaining challenges. So despite these improvements that have clearly improved the therapeutic index of the Tricyclo, there are still a lot of challenges that remain, especially in a difficult pathology like Duchenne muscular dystrophy. And beside the optimization of the antisense oligonucleotides itself, like you can play on the chemistry, on conjugation, it is actually also possible to address some of these challenges at the cellular level. Of course, some challenges are very common to all antisense oligonucleotides and even all RNA therapeutics. And these include improving the cellular uptake or improving the endosomal escape that you can actually do by co-injecting sometimes additional compounds. And we did actually a study with the Tricyclo co-injecting compounds that were facilitating endosomal escape, and that indeed improves the potency. But I want to focus today on more specific challenges that are associated with DMD. And one of them is the limited amount of pre-mRNA target that is available in Duchenne. So in the specific context of the exon-skipping therapy, this challenge is maybe less recognized as the others, but there's actually a very limited amount of target mRNA. So transcriptional studies have shown several years ago already that there were reduced levels of dystrophin mRNA when a mutation is present. And the field has long assumed that this reduction was mostly due to the nonsense-mediated decay that would break down the transcript containing premature termination codon. But recent work from LUMC, so from Annemieke Ertsma-Roos and Peter Spitali group, have shown that the NMD does not specifically affect the mutated DMD transcript, and that actually the decreased level of the transcript was mostly due to a very strong 5' 3' transcript imbalance. And this is something that we have also shown. So this is an example in the dystrophic mouse model, the MDX, where you can look at the transcription, the level of transcript along the gene at the different exon junction. And if you compare the level in wild type and dystrophic animals, you can see that at the 5' of the gene, they are quite similar, but then they start to decrease quite significantly, and towards the 3' of the transcript, there's clearly less transcript, and this is true for almost all the tissue that we've looked at. There's clearly less transcript available. So less transcript means that there's not as many transcript available for the antisense oligonucleotides to play its role. And some of the work that this paper shown, so in LUMC, was doing chromatin immunoprecipitation, showing that there was actually increased level of repressive histone marks in the dystrophic animals. That indicates that chromatin conformation would be less prone to transcription in these animals. So that raised the hypothesis that maybe if we could revert this, there was a possible of increasing the amount of DMD transcript, and this may be possible using HDAC inhibitors. So we investigated this hypothesis together with the group of Annemieke and Pietro in Leiden, trying to increase histone acetylation using histone deacetylase inhibitor to correct this imbalance and eventually have more pre-mRNA available. So we evaluated different HDAC inhibitors in co-treatments with the Tricyclo. So we selected givinostat, valproic acid, or EX527, and treated the mice for 4 weeks with this cocktail. And we're able to show that indeed the co-treatment with HDAC inhibitors such as givinostat or VPA increases the level of dystrophin restoration. You can see here, for example, in the diaphragm here, with the treatment with VPA, we almost not double, but it's like 1.7-fold of dystrophin increased expression. And if we looked at the 5' 3' imbalance, we were able to see, for example, in this diaphragm, in red here is the curve, is the level of transcript in the mice treated with ASO and valproic acid, that despite the slope being quite similar, so we haven't corrected the transcript imbalance, the levels are higher. So the HDAC inhibitor treatment has increased the transcriptional outputs and probably translates into a more efficient exon skipping and more efficient dystrophin restoration. So based on this result with the VPA, with the valproic acid, we did a longer treatment in order to confirm this effects on dystrophin restoration and eventually functional outcome. And a 12-week treatment indeed show higher exon skipping in all the muscle evaluated and higher dystrophin restoration. In this case, it was almost twofold everywhere. This was also shown by immunostaining here, where it's very evident that there's more dystrophin expression in the heart, for example. And that leads to higher functional improvements, which is shown here. If we look at the biomarker like myomycin-3 in the serum, which is already improved with ASO alone, but with ASO and valproic acid, it is even further improved. And similar with a functional test here, a motor function with the inverted grid test, where the mice treated with both compounds can hold to the grid longer. So there's clearly a potential for these combined therapies, and HDAC inhibitors together with ASO are quite promising to potentiate the effects of the oligonucleotides. This is particularly interesting these days since some of the HDAC inhibitors, such as givinostat, has been approved for the treatment of Duchenne muscular dystrophy. So we can imagine that this type of combined therapy could be quite rapidly translated to the clinic. So for the last part of my talk, I wanted to focus on the CNS part and the possibility of addressing DMD-associated comorbidities in the brain. And for this, I want to come back to some of the properties of the tricyclo that we had identified already in the earlier studies, where we had seen that the tricyclo were able to cross the blood-brain barrier at low level. So when we initially compared the tricyclo with the 2' ormethyl and the PMO, and we looked in the brain of these animals that were treated by intravenous injection, we had noticed some exon skipping only with the tricyclo in the hippocampus, cortex, and cerebellum. And that was leading to some dystrophin restoration, quite low restoration, but still here, and by immunostaining as well, we were able to detect. And what was interesting, so dystrophin is expressed, as I mentioned at the beginning, in the brain, in particular in the pyramidal neurons of hippocampus and amygdala, also in the Purkinje cells in the cerebellum. And what is interesting is that the lack of dystrophin in dystrophic animals is associated with a degree of cognitive deficits, and the mice actually display some phenotypes. One of them is enlarged emotional response that we call the unconditioned fear response, that has been very extensively studied by our collaborator Cyril Vaillant at Neuropsy. And so this is a very robust phenotype where you can submit the mice to a very mild stress just by restraining the mice for about 15 seconds and then releasing the mice in the cage. And while the video capture for 5 minutes, you will see that the dystrophic animals freeze in the cage because of this pathological stress that the restraint has induced. And this is not a phenotype that's linked to motor function, because if you only take the mice and videotape it in the cage, there's no difference with the wild type. It would still explore the cage. It's really due to this mild stress that is induced. And so you can quantify this freezing, and we were able to show in this initial study with the tricyclo that when the mice were treated with a high dose of tricyclo, they were actually, this phenotype was fully reverted. And the mice didn't freeze, or they were similar to wild type, as opposed to the other chemistry that didn't penetrate the brain. So this was very exciting and promising findings at the time. First of all, because they suggested that some of these neurobehavioral outcomes may be reversible. But of course, we wondered if we could eventually restore some other neurobehavioral and cognitive outcome, in particular, some long-term deficits that had been described in dystrophic animals. If so, what levels were required, and was there a minimum level? And of course, what would be the impacts of the other dystrophin isoform? Because as I mentioned, there are several dystrophin isoforms expressed in the brain. So we addressed this question together with a large consortium, a European consortium called BIND, so for brain involvement in dystrophinopathies, that was led by UCL and Francesco Montoni. And so this, the objective of this consortium was to better understand the brain comorbidities, the linked to dystrophinopathies, to eventually guide future treatment development. So as I mentioned, dystrophin is expressed in the brain as multiple isoforms. So in particular, the full lengths are expressed, the DP140 and the DP71. And the absence of one or several of these isoforms lead to complex phenotype. There's approximately 50% of DMD patients that present comorbidities, and they include intellectual disabilities, autism, attention deficit disorders. So they can be very variable. And one of the questions was, are these phenotypes eventually reversible? So we initially had worked with the MDX mice that I've presented you before. So that has this stop codon in exon 23. So this mouse model lacks only the full length, the DP427 in the brain. And we first did a dose response using the Tricyclo-DNA antisense oligonucleotides. So the last generation that I've presented, the PALM tcDNA PO. So we injected the oligo and saw a dose response in terms of exon skipping in cerebellum, cortex, and hippocampus that could reach about 30% of exon skipping in hippocampus. Here we also detected the effect using base scope by immuno in situ hybridization. And you can see the red dots here correspond to the skipped to the exon 23 skip junction. And so we were able to see clearly in the granular layer of the hippocampus that there was a strong exon skipping. This was associated with dystrophin restoration, again, dose dependent and with a higher rescue in the hippocampus. And this also correlated with this improvement in the fear response that I've just presented. And clearly, the more dystrophin we had, and the better the phenotype was improved. So we had a significant improvement of the fear response with the high dose of tricyclo injected. So we next moved to a different mouse model, which is the MDX52 mouse model, which lacks several isoforms of dystrophin in the brain, in particular the full-length DP427 and the DP140. And so this is similar to approximately 60% of patients that lacks both isoforms. So we had shown that this mouse model presents enhanced anxiety and also severe impairment in fear learning. And molecularly speaking, this is also an interesting mouse model because the deletion of this exon 52 is amenable to two different possibilities of exon skipping. So you can either induce exon 51 skipping, and in that case, you would restore only the DP427. And this is because the start codon of DP140 is actually included in exon 51. So clearly, if you exclude that exon, you cannot restore the expression of DP140. Whereas if you skip exon 53, which also restores the reading frame, you will preserve the start codon of the DP140 and eventually be able to restore both isoforms. So we started with exon 51. You may wonder why, but this is because it actually represents the largest proportion of patients that are eligible for exon skipping therapy. And also because we wanted to see in this mouse model if what the rescue of the DP427 only would do. So we started with an optimization of the ASO delivery, and within the consortium, we had access to different chemistry. So we compared PMO to prime-methoxyethyl and tricyclo after direct intracerebroventricular delivery, because we wanted to reach very high level of exon skipping to start with, to then eventually start decreasing the level and see if we had a minimum dystrophin. So that's why we went to local delivery. So here I'm presenting you the results of the direct comparison of tricyclo and PMO. Well, you can see that in both cases, we had the ICV delivery induce a quite widespread biodistribution of the compounds. So we were able to see a nice distribution of either of the oligo. The levels were always higher with the tricyclo. And we also compared different administration methods in the dystrophic animals, trying to increase the levels. So we compared single ICV delivery to repeated ICV delivery by the insertion of a cannula in the mice. Unfortunately, that didn't improve the level of exon skipping, and it was quite similar to a single administration, which is probably due to the anesthesia that's used for the subsequent administration, which is a much lighter anesthesia, and that plays a role in the diffusion of the oligo. And we also compared with an administration of oligonucleotides using some mini pumps. So osmotic pumps are implanted at the back of the mice, and that can deliver the oligonucleotides through a very long period. So for example, here was about over 2 weeks. And in that way, we can inject even higher amounts. So we even went up to 2 milligrams of oligonucleotides, but actually it turns out to be very variable depending on the animal. So we had one mice that was responding extremely well, but most of the time we obtain relative low levels. So in the end, we concluded that the most reproducible injection methods that was leading to the highest level was the bolus ICV injection. We had also actually compared intracisternal magna in this paper and really showed that ICV was the most efficient. So based on these injection methods, we then investigated the extent and the duration of the dystrophin restoration over time. So we did so the ICV injection of the Tricyclo targeting the exon 51 in the brain of MDX52 mice. Looked at the biodistribution of the compound, again, very widespread distribution across the different brain structure. You can see that with time, the level of compounds clearly decrease. So this was expected. So a very strong decrease between 3 and 11 weeks. Interestingly, in terms of efficacy, the levels are quite stable. So between 3 weeks and 11 weeks, the levels are about 20-25% of exon skipping. And this is interesting because it suggests that the productive fraction of oligonucleotides is actually stable, and the part that is eliminated and the part that's decreased is likely the unproductive fraction. So we also validated the dystrophin restoration and the extent and duration of the restoration. So by Western blot, we detected between 3 and 15% of DP427 in the different brain region. That was again stable between 7 and 11 weeks. The DP427 expression was also confirmed by immunostaining. So dystrophin is normally expressed here as a punctuate staining in the inhibitory synapses of the pyramidal neurons. And you can see that in the treated animals, we could definitely detect that punctuate staining. Locally, in the hippocampus, we went up to about 30% of restoration. And that validated, again, the therapeutic window for behavioral assessment between 7 and 11 weeks. So we could then inject larger group of animals to start the behavioral analysis. And so we started with the unconditioned fear in treated animals with the oligo targeting the exon 51 compared to a group of animals treated with a control oligo. The good news was we could find the same phenotype even in mice that were subjected to surgery, and the oligo control had no effect on that strong phenotype of the freezing response. So the mice treated with the Tricyclo exon 51 expressing dystrophin had an improved unconditioned fear response. We also looked at anxiety that is impaired in these mice using different tests. So for example, here the elevated plus maze, in which you can detect the anxiety by the mice not willing to go and spend much time in the open arms. So this is what is shown here. So the dystrophic mice treated with the control spend less time and go less. So there's less entries in the open arms, and this phenotype is actually restored after the treatment. You can also evaluate the anxiety using light-dark, the choice test. And in that case, the anxiety is shown by the mice not willing to spend time in the lit compartment, which is very anxiogenic for them. And so they do not spend much time in the lit compartment, in contrast with the treated mice that were not statistically significant from the wild type. And finally, we looked at the condition fear memory. So this is an auditory cued fear conditioning test where there's a learning phase for the mice to learn that the tone is going to be associated with a shock. And so in the learning phase, we were able to show that the treated mice in red were actually very similar to the wild type. So whereas the control mice show a delay in learning this particular phenotype. And you can then assess the memory and the retention for this test. And 24 hours later, where the treated mice were actually at the same level here, showing that they can kind of remember that the sound is associated to a shock. But you can see that the phenotype is not fully restored because they do not recapitulate the full phenotype of the wild type mice. So there's only an improvement in the fear memory. So overall, this showed that the partial rescue of the DP427 improves anxiety, fear learning, and fear memory in these mice, which is important to suggest that this may be reversible in patients. And the last part that I wanted to show you was some work that we're currently doing because we're wondering if we could eventually restore more neurobehavioral phenotypes if we could induce restoration of dystrophin earlier and do an early postnatal treatment. So we've investigated ICV injections in neonates. And so we're doing ICV injection at P0 or P1 and look at the dystrophin, exon skipping and dystrophin levels at 4 weeks, 7 weeks, or 14 weeks after the treatment. And you can see that this leads to very high levels of exon skipping and also of dystrophin restoration. So, for example, here, 7 weeks after the treatment, we are about 20% of dystrophin restoration, which is higher than what we had by treating the adults. Unfortunately, the levels decrease with time. And so in the time where we wanted to do the behavioral assessment, we have a decrease. That means that we would need to re-inject the mice ICV again, which it's something that we can do, but we wanted to investigate eventually an alternative method that would induce a longer-term restoration. And for this, we turn back to an old tool that we've developed nearly 20 years ago now, which is a vectorized approach in which the antisense sequences are inserted into the U7 snRNA carrier, and the U7 snRNA modified with the antisense sequence is then vectorized into an AAV vector. We had previously shown that this is a very efficient system that allows the colocalization of the antisense sequences with the pre-mRNA, which is very stable for the antisense sequence and induces a long-term effect, at least in muscle. So we've injected this system. So the U7 was made for the exon 51, and it was injected ICV in the neonates. And similarly, we looked at different time points. And by doing so, we've also detected very high levels. And in that case, the levels were still increasing with time and not decreasing at all. And this is also true for the dystrophin restoration that even reached around 50% of restoration in the hippocampus, for example. So this tool really allows us to have a high and a long-term expression of dystrophin that's very appropriate to then do all the behavioral studies that are currently ongoing. So to summarize this talk today, I've shown you first that the pan-tricyclo PO, so the full phosphodiester, a promising chemistry for the treatment of Duchenne muscular dystrophy. So it is currently evaluated in DMD patients by Sky Therapeutics. Regarding the CNS parts, we've shown that the direct intracerebroventricular injection of antisense oligonucleotides allows a widespread exon skipping in the CNS of dystrophic mice, and that even a partial rescue of the full-length dystrophin can improve anxiety, fear learning, and fear memory in the MDX52 mice, which is important to suggest that these deficits may be reversible in patients. So there are more investigations needed, not only to look at earlier postnatal, but also to investigate the additional restoration of DP140, and all this is ongoing. But yet these are encouraging findings, especially at a time where new generation of antisense oligonucleotides are being developed, and some may offer the possibility of crossing the blood-brain barrier and therefore treat both the muscular phenotype, but also the comorbidities that are associated with the lack of dystrophin in the brain for these patients. So I just want to finish by thanking all my group, and some of them are here in the lab, and in particular for the brain studies, I want to thank Ophélie Vaca and Mathilde Doisy, but also some of the former members, Amel Saoudi and Fauzi Zarouki, who had initiated that work, the BIND Consortium, and our collaborator Cyril Vaillant at NeuroPsy, and Francesco Montoni for leading this great consortium. Of course, Sky Therapeutics and all our supports. And I'm happy to take any questions.
Thanks, Aure. That was a great presentation. Let's start with the question. So it's always puzzling for me. So and always, I think always the first question that comes in mind is how much restoration. So I'm a brain person, so very interested in the brain. So how much restoration of the dystrophin in the brain is needed to get a complete rescue of this particular phenotype? Because I think in your graphs you showed 5 to 10% increase. So is it enough? Do you think that might also be enough in patients? And then the follow-up question: can it also be too much?
That is a very good question. So we don't know. So there's, I mean, this has been a long-term question in the muscle field, and now I think in the muscle we kind of all agree on an eventual percentage of between 10 and 20%. I guess in the brain it's still very new, and we don't know how much dystrophin is needed. From our earlier studies, we were able to show that after systemic delivery, even when we had only 2-3% of dystrophin, we had a full rescue of some phenotype. And in subsequent studies, we've shown that with 10 to 15, it's only partially restored. So we don't know yet how much is needed. We don't know if it's a question of level of amount of dystrophin rescue, but also which isoform and also timing. So this is why we also try to do early postnatal. So I guess it's a combination of, timing, localization, amount of dystrophin. So all these questions remain open. I guess the promising and encouraging finding is that even with a relatively low partial restoration, such as 5-10%, we are able to restore some of these phenotypes, some of them fully. So I think that's quite encouraging.
Yeah, and then the follow-up question would be, so which cell types in the brain are important? So in which cell types do you want to have this rescue? Are some cell types more important than others, or do you just want to treat the entire brain?
So at the moment, we do want to treat the entire brain because, and this was one of the, an additional objective of the BIND consortium was to better characterize exactly which cell type the dystrophin is expressed, and not only which dystrophin isoform is expressed. So we know that the dystrophin at the full length is expressed in inhibitory synapses in the pyramidal neurons, in the hippocampus, in amygdala, also in Purkinje cells. So there's quite a broad expression of the dystrophin. So that's why we want to have the wider expression as possible. The good question that I didn't reply, I forgot, is the about the overexpression. We don't know either, and that's an interesting point because in some of our strategies we are also using a gene therapy, so AAV gene therapy, trying to restore some particular isoforms that can fit into an AAV. And in that case, it tends to induce overexpression. And so we don't know yet if eventually an overexpression of the dystrophin could eventually lead to detrimental effects.
I think it's a bit noisy, so I mute myself, try to mute myself. So if you have a question for Aure, please use the Q&A function. So I will start with the question from the Q&A. So first of all, it's more a general question. So are all in frame mutations lead to functional dystrophin protein? So if you have an in-frame mutation, so you have a different combinations that you can make, right? So is it
so not necessarily. It can depend on the segments of the protein that you. So there are parts of the protein that are really essential. So even if you have an in-frame deletion in that part of the gene, if it deletes an essential part of the protein, like the actin binding domain, so in that case, the protein won't be functional. And this is why we always say that the exon skipping is applicable to a large part of the patient, but it's not all patients that are eligible. So some particular mutation are not eligible to exon skipping for that reason.
Have you tried other lipid moieties besides the PALM? And do both the 5' and the 3' conjugations work, or is there a preference for one of those?
Yeah, so we've indeed tried several fatty acids, and so I think that most of them have also been tested for other compounds, and I'm also thinking of a study published by the IUNIS group with the C8 gapmers. And so we tested most of the similar fatty acids. And the PALM and the C18 were probably the most efficient. So we selected the palmitic acid because it was the best one. We had tried also the 3', and they are similar, but from a synthesis point of view, it was easier from the 5'.
Yes, another question. So the current therapies for exon 51, they exhibit greater skipping profiles in skeletal muscle compared to the heart. In the non-human primate data that you showed, it seems to be the other way around. Any thoughts about this?
Yes, so it's true that the Tricyclo have always had a very strong tropism, if I may say, as it's a vector term, but they tend to target the heart very well. And this is as opposed to the other chemistry, and we always thought that it was due to their hydrophobicity, so they were more efficient in the heart, really due to their hydrophobicity. But we don't know exactly why, but they are definitely going into the heart even more than the skeletal muscle.
Question from Hans. Excellent presentation. Have you looked at the efficacy and safety of fatty acid ligands of different lengths?
So we didn't push forward many fatty acids. So when we did the studies in rodents, identified relatively quickly that palmitic acid was the best one. So we really pushed that one forward. We didn't do all the safety studies with the other fatty acids. So I wouldn't be able to comment on this.
Question from Mark. So, were there any toxicity seen after the ICV delivery of the tricyclos? And how did the safety profile compare with the existing treatment strategies?
So we were able to detect some toxicities when we injected really high dose. So as I mentioned, we went up to 2 milligram. So in that case, the mice didn't tolerate the oligo very well. But for mice, I think that's going really over the top to inject 2 milligram. Otherwise, in the study that I've shown, there was no particular toxicities observed if we keep the dose low.
So that's the next question. What's the maximum dose? Do you have any idea about it?
Yeah, we are limited with in a way, the solubility of the stock that we receive. So our stock is at and the limited amount, the limited volume in mice that we can do ICV. So that's why we always up at 400 microgram, because that's the maximum dose we can inject with the solubility at the moment.
And how easy?
Except when we did the cannula for repeated, or the osmotic pump, and in that case, because the volume was higher, we were able to inject higher, but it turned out to be less effective because of the. So I think the bolus has it really, and the anesthesia also changes the CSF flux. Which facilitates the uptake, as opposed to a light anesthesia, or even administrating when the mice is awake once you have inserted the cannula.
Yeah. So, how easy is it to make big amounts for clinical trials? And can you also comment on the cost if you compare this versus other cancer therapies?
Okay. Well, how easy to make? I guess now it's made. I mean, it's like all the. I mean, it's done by a CRO, so now that they have the process, they can make it easily. And the cost, I wouldn't be able to comment. It's probably a little bit more expensive than typical chemistry, but I don't know.
Okay, question from Julia. Great talk. So your blood-brain barrier crossing data is very interesting. How old were the animals injected in these experiments, and what percentage of the oligo is crossing? And what do you believe the mechanism is?
Okay, so yeah, it is a great question that we've tried to answer for many years. At least regarding the mechanism. So all of the mice, they are adult. We've treated mice basically between, I don't know, 6 and 12 weeks of age, and it crosses. The percentage of oligo that's crossing, it's a very minimal part. It's difficult to judge because we're doing multiple injections, so it's always repeated weekly injections. I've never calculated, but it must be like under, and yeah, I don't know. I'll do the calculation. I'll tell you, Julia. But and the mechanism, we haven't been able to identify a specific mechanism so far. So it's probably linked to the hydrophobicity of the compound. It's really linked. I mean, it's definitely linked to the tricyclo, not to the fatty acids. When we use the fatty acids now, it didn't change the property, but it didn't improve it either. So it's definitely not due to the palmitic acid. It's really due to the hydrophobic nature of the tricyclo. Whether it's due to the capacity of forming nanoparticles, because we had shown in several years ago with our collaborators from the Karolinska Institute that the tricyclo were able to self-assemble into some sort of nanoparticles. So can it be due to these particular properties? We do not know.
Okay, so we are almost reaching the time. So it's still a lot of open questions. So if you have a question for Aurélie that is not answered yet, you can send the question to the OTS. So info@oligotherapeutics.com. com.org. You can find it on the OTS website. So one final question. So I think you showed the comparison between the tricyclos and the PMOs, but you did not show any of the two PMO data. So what about that? What happens with the data?
So at least in the context of DMD, they were less efficient. They were, I think, even a bit worse than the PMO. And in order to induce, so the 2' methoxyethyl experiment were done by the group at LUMC. That was also part of the consortium. And they had to inject a much higher dose to be able to detect between 5 and 10% of exon skipping. They were up to, I think, at least 5 times of injection, 5 times of 400 or 500 microgram to be able to see 5 or 10%. So clearly it was much less than having 20 or 25% with only one injection of 400 microgram. So they did not work very well.
Good. And Aure, thank you very much for your excellent presentation. I enjoyed it a lot. So the next webinar, we will update you via the email, and see you in a few weeks. Bye-bye.
Thank you.